Hello again, everyone. Welcome, and thank you so much for joining us for the Congenital Muscle Disease Tissue Repository Invited Speaker Series. We will be recording this presentation. To reduce the distracting noises, we ask that audience members please click the mute button on your device during the talk until we get to the Q&A point of the presentation. And I think Sandra will be pausing at various times to take questions. She'll let us know. And if you have called in using your telephone, please make sure that your telephone microphone is muted until the Q&A portion of the talk. Okay, so just to begin, a little bit of an introduction. My name is Stacy Cassette, and I'm the manager of the CMD Tissue Repository, as well as being curator of the CMD International Registry. I also coordinate sample donations to the CMD Biobank for the development of cell lines. I've been doing this work since about 2012 and had worked in other aspects of medical research for about a decade before coming into this specific role. As the parent of a young lady with a congenital muscle disease, I knew that it would be up to us to drive the research forward in order to find solutions to these life-limiting conditions. So our mission at the CMD Tissue Repository is to provide a free service to CMD individuals and families who are interested in supporting medical research and treatment options through tissue donation. We then make those tissue samples from patients available for research and treatment studies. Centralizing the biological samples and the data in this way makes it easy for scientists to request and obtain what they need to expand our understanding of these conditions and how to treat them. Plus, it makes things a lot simpler for families to contribute when the opportunity arises, such as through muscle biopsy, another type of surgery, or end-of-life procedures. As of the end of 2017, our tissue donors had contributed over 370 tissue samples that are now available for research use, which is really amazing. Our success depends on the support and participation of the community members, so I want to extend a warm thank you to everyone who has participated and who continues to advocate for finding solutions for these congenital muscle disorders. As part of our education and outreach goal, we host webinars on topics of interest to the CMD communities. If you are a Facebook user, you can go to our page to like and follow. And if you're not a Facebook user, another way that we can keep you informed is through your registration with the CMD International Registry. And I did put that website up here, as well as the website for our tissue repository program. I can be the central contact person for really any of these three programs. And anyone who hasn't registered yet with the CMDIR, please take a moment to go there and register yourself or the person you care for who has CMD. It's really important that we get an accurate count of the people who are affected and how their particular disorder is progressing or stabilizing. It's really good information for the scientists to have. And we do provide that type of information de-identified at the request of scientists. So this is Dr. Michael Lauer. He's the director of the CMD Tissue Repository that I manage. He's also the associate director at MCW's Neuroscience Research Center. He's the director of pediatric pathology, clinical neuromuscular lab. He's a board certified physician in anatomic pathology and neuropathology, the associate professor in the Department of Pathology at the Medical College of Wisconsin. And he's a noted expert in congenital muscle disease. And at the bottom, these are some of the supporters and individuals who have started foundations that do support the Congenital Muscle Disease Tissue Repository. Today, I am delighted to have Sandra Donkervoort with us today as the guest speaker. Sandra is a board-certified genetic counselor working at the National Institutes of Health, the Neuromuscular and Neurogenetic Disorders of Childhood section. Sandra graduated from Sarah Lawrence College in 2007 and joined Karsten Bönnemann's team at the NIH in 2011. Her research is focused on identifying the underlying genetic cause in patients with unknown neuromuscular disease. Welcome, Sandra, and thank you so much for presenting. I am going to switch over the presentation role to you. Just give me one moment.
Okay, thank you so much, Stacy.
You are welcome.
I think my screen's up, I think. Perfect.
Yes, it is. I see it.
Okay, great. Thank you. All right, yeah, so today I wanted to just give a brief overview about genetics and obtaining a genetic diagnosis in congenital muscle disease. This can be very complicated, can be a very long process for some patients. So I just wanted to give more of an overview of the different steps that can lead to confirmed genetic diagnosis. Overall, you know, let's see if the slides, I don't think my slides advance. Oh, there you go. All right, so just a brief outline, just to make sure that we're all using the same language and the same terminology. I just want to review some basic genetic concepts, go over some of the inheritance patterns that can be involved in our with the muscle diseases, specific genetic testing strategies, how do we interpret the genetic testing data that comes back, the different variant interpretation, the things that are on your genetic testing report, and then briefly touch upon clinical versus research-based testing. So overall, the presentation is going to be, of course, general, with general concepts about genetics and approaches to genetic testing. So I understand that everyone that's on the call may be at a different step of their child or their own genetic odyssey and genetic path. Some may have a genetic diagnosis, others may not. So hopefully throughout the presentation, I can answer some questions. I'll pause at different steps to see if there are specific questions about the things I discussed. And if there's more individual or case-by-case questions about your test results or your child test results, I'm happy to maybe discuss that in a separate call to discuss individual cases and individual results. All right, so let's see. It's a little bit of a delay with the slides. There you go. This may bring back memories from high school genetics and high school biology classes. But just real quick, that we're all on the same page, Genetics 101. We are all made up of billions and billions of cells. Within these cells, in the center of the cell, in the nucleus right here, that's where we contain the majority of our DNA, our genetic information. DNA really is a genetic instruction code. It's a set of letters that has to be in a specific sequence for the body to read in order to generate proteins. So we have our DNA in the nucleus. It's a strength of letters, and the body reads that to make proteins. Proteins really are the building blocks of our cells, and they're required for proper development really early on, even during pregnancy, then after we're born, for the structure, function, and regulations of all our tissues and organs. The specific instruction code is packaged in a gene. So one specific gene will code for one specific protein. And the genes are located on structures that we call chromosomes, that are these bigger structures. Each chromosome can therefore contain many, many genes, and we inherit one chromosome from our mother and one chromosome from our father. Let's see. Hey, Stacy, do you know? Oh, Yeah, the slides are really slow on this end. So when we look at our chromosomes, when we take a picture of the chromosomes, this is what they would look like if we line them up nicely. So we have 22 regular chromosomes, autosomal chromosomes. And they come in pairs, as I said, one's from mom, one's from dad. And then the last set are the sex chromosomes. So XY would be a boy, and then 2 X's is for a girl. So every chromosome has a gene, so genes also come in pairs. So what are genetic mutations? What is variations in our genetic information? So a mutation is a permanent change in the DNA sequence of a gene. So it's basically a letter that's changed or missing or repeated. It's a difference from what it should be. Many of these variations are very, very common. Most of them are harmless and do not cause disease. Later on, we'll touch base a little bit more about how do we interpret these genetic variations and when do we know whether they're associated with disease and when do we call them benign and harmless. When the code is altered, so when the genetic code is altered, it can lead to either no protein or an incorrect production of protein, and that may have downstream effects and lead to disease. So this is what a mutation could look like. So you have our letters here, and either there's a misspelling, what we call a missense mutation from an A to a G, for example, or there's a deletion where letters are missing, or in some situations there could also be an insertion. All of these situations could lead to either no production or incorrect production because the instruction code is altered. Typically, these mutations are present at the time of conception, and they're found in every single one of our cells throughout our body. These mutations can happen spontaneously at conception, what we call de novo, or they can be inherited through family from generation to generation. So very often, or when families would say, I have the gene for blah, blank. Really what they probably mean is we all have 2 copies of every gene, but they probably mean we have a mutation in the gene that causes a specific disease. So there's different ways in which a mutation can run in families. The 2 most common types are autosomal recessive inheritance and autosomal dominant inheritance. So these have nothing to do with the sex chromosomes, equal between males and females. In an autosomal recessive condition, which you can see here, each chromosome, one gene on the chromosome has a mutation. So here is the dad, one gene has the mutation, the other gene has a normal sequence and normal instruction code. Same with the mom. Then because there is the backup copy, the healthy copy here, there is enough protein being produced for the body to function without any problems. 50% in this situation is enough for healthy development. However, when this couple has children, there's a 1 in 4 risk that the child inherits the copy with the mutation. So the dad can pass down this copy and the mom passes down this copy, and the child could be affected with disease. This is recessive inheritance. The child does not have a backup copy anymore. Other scenarios are that children are carrier, or the child inherits 2 of the normal copies. So this is recessive inheritance. And this could happen, there would not be any family history, so you meet with a genetic counselor, they would trace the family back, ask all these questions about parents, grandparents, no one else is affected, but these mutations silently can travel through the family, and you only find out about it because partner also is a carrier. The scary thing here is everyone carries more than a handful of these potentially disease-causing mutations. We just would never know about it unless we meet with someone who's also a carrier and happen to have an affected child. The other common scenario is autosomal dominant inheritance. So this is different. So in this situation, 1 copy of a gene with the mutation is enough to cause disease. In this scenario, often what happens is that this copy actually has a toxic effect, and because of that interferes with the healthy copy, leading to symptoms. If someone is affected with a disease that is transmitted in a dominant fashion, there's a 50-50 chance that future pregnancy and future children are affected. Either the affected individual passes on the healthy copy or the copy that has symptoms on, the healthy copy or the copy with the genetic mutation. A variation of autosomal dominant inheritance is de novo inheritance. So this is a new mutation, meaning that both parents have the normal allele with the normal sequence, but a mutation spontaneously occurred at the time of conception when the egg and the sperm meet. These mutations, these spontaneous mutations, are very, very common. Usually they occur in parts of the DNA that are not clinically relevant, but if they happen in the gene, it's associated with symptoms, and if the change is severe enough, the child could be affected. Once the child has this mutation, so once it's established in the child at conception, it's present in every single one of the cells, and then future generations would have a 50-50 chance of inheriting the mutation as well. Other siblings are not at increased risk. This is something that happened in the patient first. Then finally, this is not as common, but this is for X-linked inheritance. We see this in, for example, Duchenne muscular dystrophy or FHL1 dystrophy. This is a mutation that affects a chromosome, one of the sex chromosomes. So there's a genetic change on the X chromosomes. As I said earlier, females have 2 X's, so they always have a backup copy to compensate for a change on the other chromosome. But then if this mom has a boy and the boy inherits the Y chromosomes from the dad, the boy does not have a backup copy and may show symptoms. Carrier females may or may not show symptoms depending on the disease. And then of course, there's always the chance of inheriting the healthy allele. So this was really genetics 101 in a nutshell. I'm not sure if there's any questions right now just about these basic genetic scientific concepts. I'm happy to take them. Otherwise, I'll just continue. Okay, perfect. So why is it so hard? What makes genetic testing so complicated? Really, it's because we're dealing with a huge amount of information. Right. There you go. Apologies for that. The Human Genome Project was completed in 2001, and this was the completion of the complete sequence of the individual human genome. So it's all of the letters that make up our DNA, which is 3 billion base pairs of DNA. So 3 billion letters that are instruction codes. To put that in perspective, if you could type 60 words per minute for 8 hours a day, it would take approximately 50 years to type the human genome. So it really is a huge amount of data, and we're just starting to grasp the clinical implications of this. We don't, even though we know the sequence of the letters, we don't fully yet understand how this sequence leads to proteins and how this leads to our body functioning correctly. Of the whole genome, we have about 20,000 genes that actually code for proteins. So 20,000 packaged instruction codes that are needed for building and generating these proteins. That's only about 3% of our complete DNA sequence. So there is the majority, 97% of DNA, initially we thought was junk DNA. They don't code for proteins, so we thought that may not be clinically or evolutionally relevant anymore. Now that we're learning more and more about this supposedly junk DNA, we're finding that maybe changes in these non-coding genes could also be clinically relevant. So there's different types of genetic testing that can be done. Most importantly, which happens commonly, first step in the clinic is the diagnostic testing. So diagnostic genetic testing is to identify or rule out a specific genetic disorder. That's suspected based on physical signs, symptoms, clinical history, and in our patient population also often muscle biopsy findings. Once the diagnostic testing has confirmed a genetic diagnosis, additional testing is available, such as carrier testing. So this would be done in the setting of recessive inheritance to see which parent carries which mutation, and then that information can be used for other siblings as well, or for other family members if interested. Other types of testing, for example, if the disorder is of adult onset, so it starts later in life, once it's diagnosed in one individual, other first-degree relatives, if they're interested, could pursue predictive and pre-symptomatic testing to see whether they inherit the same gene mutations and may develop symptoms in the future. And then also, once a genetic diagnosis is established, families can use that information for family planning. Prenatal testing becomes an option, which can test a pregnancy. And then another option in some families may be pre-implantation genetic diagnosis, which is a combination of IVF and genetic testing to only potentially transfer embryos who tested negative for the genetic disease that was identified in the family. So those are the different types of genetic testing. Just to keep in mind, all of them depend on having a confirmed diagnosis in the affected individual first. So there's different types of strategies. There's many different types of testing that can be done. We have, first of all, karyotype testing. Karyotype testing takes a picture of the chromosome and lines them up nicely to look at whether or not there's any chromosome abnormality. So either an extra chromosome or big chunks of chromosomes of a chromosome that's missing. This is commonly done as a first test at birth, just to make sure that there's no really significant chromosome abnormalities. Another test that can be done is microarray testing. A microarray will look at all the chromosome regions to make sure that they're present and that they're at the right spot. So it can see whether a portion of the chromosome is deleted, extra, or maybe switched at a different spot. And then there's the sequencing. So the genetic sequencing reads through every letter of the gene to find whether there's any changes in the gene that may cause disease. So either a misspelling, what we call missense mutation, or whether small portions of the gene is missing or extra. Larger deletions or duplications can only be picked up through deletion duplication testing. So that's, again, another genetic strategy that can be done to help with the diagnosis. So when it comes to sequencing-based testing, there's different kinds of testing that can be ordered, and this may vary on a case-by-case basis. Sometimes it's a stepwise approach where people start with single gene testing and then move all the way up through research-based genome sequencing. And other testing, a gene panel may give the direct answer. It's a case-by-case basis depending on an individual's presentation. So targeted gene sequencing, very simple. It's when a patient comes in and there's one gene that has a very, very high likelihood to be causative of the symptoms. that one gene can be sent off for testing, and the answer can be received relatively fast. And that answer would be yes or no for a disease-causing mutation in that one single gene that was tested for. In some situations, this can be done because there's a high clinical suspicion that this gene is causative, just because the patient presents with symptoms that are very typical for this specific gene. Could also be because, for example, muscle biopsy was done and the lab looked at specific proteins and a specific protein was missing. So we can just test the gene that we know is responsible for that protein to get an answer. So that could be a relatively fast way in getting a yes or no answer for that one specific gene that's tested. Then in some situations, it's decided that it's better to take a little bit broader approach and pursue gene panel testing. So a gene panel is a panel of a pre-selected number of genes that have known associations with the clinical symptoms or the differential that's being considered. The gene panel is usually tailored towards a specific disease category. So it could be a panel for dystrophy or a panel for myopathy or cardiac disease. It's important to keep in mind that the lab is the one, the genetic testing commercial laboratory is the one that determines which genes are in the panel. It's not the physician, it's not the clinician that sees the patient, it's the lab. Some panels are small, some panels are large, but they may not have, for example, all of the muscular dystrophy genes, just a handful that they were set up to do testing for. So it could be that a gene's not on the specific panel, so it's important to make sure that the appropriate panel is tested when you see a patient, when a patient's being seen. Then a broader approach in the panel testing is the whole exome sequencing. This is a relatively new test, but it's being more and more ordered in the clinics, in the MDA clinics. And instead of looking at a panel of genes, the whole exome sequencing looks at all of someone's protein coding genes. So these are all the variants that can be found in what we initially thought were all the relevant protein coding information, which is only 1.5% of the genome, yet still a lot of information to go through. Expanding it even bigger is the whole genome sequencing, which can look at the complete genome, and the RNA sequencing, which can evaluate the genetic readout from a specific tissue, and in our population, that's usually muscle. Both whole genome sequencing and RNA sequencing are still relatively new testing, and a lot of it's still done on a research basis. The data analysis is not optimized just yet. It's still, the technology itself still needs to be developed, but these are also testing that are coming on the horizon. So how do we determine which gene to test for, or which strategy to pick in terms of the genetic testing for patients? So this is really depends on a clinical history. We like to take a look at the patients. What are the clinical features? Are there other systems involvement, like do we see cardiac involvement? Anything else that stands out from the history? Additional testing can help guide us which direction to go, such as the EMG, muscle imaging. Specific disorders may have specific signs, muscle imaging, brain imaging as well. Some muscular dystrophies may have changes on brain MRI imaging. And then the muscle biopsy can also be very, very helpful to see if there's any structural changes that can guide us in the specific direction, or whether there's any protein that's missing or abnormal, and then we can kind of zone into those specific genes that we know code for those proteins. So all of this needs to be taken into account when ordering the specific testing, and then also looking at the variants that may be generated by the genetic testing. And we'll go into the variant interpretation in a little bit. So real quick, just to touch base on exome sequencing, because it really is the newest test that's more commonly used in the clinical setting. So the exome sequencing looks at all of our protein coding genes. So it can be a relatively cost-effective way to find an answer, just because you don't have to go through this process of starting with the individual gene, then going to a panel, and then going through an exome sequencing. In some situations, it can also be used to identify new disease-associated genes. So it's a technology that's also used in the research setting very often to try and find new genes that have not yet been reported or associated with congenital myopathy. Initially, when we started using exome sequencing, we were all excited and we thought this was going to provide an answer for all of our families. But it turns out, and this seems to be pretty consistent among different other disorders as well, exome sequencing is only able to identify diagnosis in about 50% of the families and patients on whom the testing is pursued. So it's definitely not a perfect test. It could be that we're missing things because of the technology, or that there's just all these unknowns in the rest of the genome that we have to further explore. The exome sequencing does generate an enormous amount of data and variants. As I said, variants are very, very common, and the exome sequencing will read through every single base pair, every single letter of our protein coding information, and we end up with huge reports of many, many variants. And this is when it's really important, again, to have a good understanding of the patient's clinical presentation and to make sure it fits with what we know about a specific variant, because having no genetic diagnosis is hard and frustrating, but it's even worse to have an incorrect diagnosis based on variants that may not be the one that cause disease. So with exome sequencing, if it's done, or if the local clinician is not as familiar with the data interpretation or the clinical spectrum of symptoms, there is a chance of an incorrectly assigned diagnosis and an incorrect label that we should be very, very cautious of. To help with the data analysis, it's always done to do exome sequencing as a trio, meaning the patient and include the parents, because then we can start tracing those variants back and see which one's inherited, which one's new in the patient, to help filter through all these variants. So as I said, the exome sequencing is not perfect. There still remains patients that are undiagnosed. Could be because of the technology. Exome sequencing does not cover every single gene 100%, so there may be what we call dropout of specific regions, and this can happen in genes that we know are associated with the congenital myopathy. So it's always good to make sure that those specific genes are ruled out, perhaps by targeted testing or a panel. And in some situations, it may not be the appropriate test. Exome sequencing does not pick up deletions or specific repeats. So if that's in the differential, so also to keep in mind, those things will be missed by exome sequencing. And then lastly, because the exome sequencing reads through every single letter or base pair of our genes, it's possible that the lab stumbles upon findings that initially they were not looking for. So they discover something by chance that's unrelated to the patient's symptoms that were being evaluated. So we call those incidental findings. And in some situations, there may be completely independent secondary findings that the lab actually will look for in also non-disease related genes, but they may report back findings, for example, predisposition cancer findings. If they find these things, they don't want to sit on it, but they want to report it back to the family because they may have clinical implications. Let's see. Before I go into variant interpretation, any questions about the genetic testing strategies discussed so far? Okay, great. So finally, this is how do we interpret these variants that come up when we do the genetic testing, and when are these variants likely? So as I discussed earlier, variants are very, very common. There's really three different categories that we can put them in. We have the pathogenic variants here on top, and these are changes, mutations that are known or thought to be directly contributing to the development of disease. We have the likely benign variants at the bottom. So these are just variants in our genetic information that are not associated with symptoms. They're probably just inherited through our family, makes us related to our family, makes us, who we are, but are not associated with symptoms. And then we have this middle bucket, the most frustrating one. These are variants of uncertain significance. So basically variants where we don't know as of yet how to classify them. Sometimes as we do additional studies, these variants of uncertain significance can be reclassified. So we may find out that there are additional patients seen by collaborators who have the same mutation, and we may not find it in healthy individual control databases. In those situations, as this other information becomes available and is being published, a variant that initially we thought was of uncertain significance, now with the new evidence, we feel confident that we can classify it as pathogenic. Or the other way around, where we see a variant of uncertain significance, and as we're doing more and more population-based testing, screening of healthy individuals, we see that actually it's relatively common in the healthy population. So then over time, we may decide that this variant is actually benign. So even though maybe a couple of years ago, genetic testing came back was inconclusive with these variants of uncertain significance, it is always helpful to once in a while check back in and see whether these variants are reclassified. This process is not fast. It can take some time just because it takes time for science to be published and science to come out, and we also don't want to make mistakes. So it's definitely not a fast process, but over time, a lot of, especially now that genetic testing has become more and more accessible, more and more knowledge is being generated. So we do see that these variants get reclassified. Sometimes it takes years, but it's important to maybe revisit that once in a while with the clinicians to see if any new information has become available. So interpreting your genetic testing results, the first one, the pathogenic variants. So these are disease-associated variants. So what we call a positive genetic test. It confirms the diagnosis. One thing to keep in mind that this is only in the setting of correct inheritance. So if a disorder is known to be inherited in a recessive manner, meaning that 2 mutations need to be present, having only one pathogenic mutation does not explain the complete picture. In those situations, it could be that there's a hidden mutation on the other chromosome. So it's important to make sure that the complete picture fits. If it's a recessive disease, 2 disease-causing mutation or 2 pathogenic mutations need to be identified. Once we do have a positive or pathogenic diagnosis, we can use that information to direct medical management, care, screening. For example, we can go back to the literature, see whether or not there's any cardiac involvement known for this specific diagnosis. We can see whether or not there's any information known about when to initiate pulmonary support to prevent certain outcomes. So the benefit of having a genetic diagnosis, of course, is you don't want to have any surprises. There do not need to be any surprises because you go back to the literature and see whether what's known about the mutation. Additionally, once we have the pathogenic diagnosis in hand, we can facilitate genetic counseling. We can provide recurrence risk information, family planning. Then more and more studies now use a genetic diagnosis as eligibility criteria for clinical trials. So it's important to have that in hand if a clinical trial becomes available. The patient, the family is ready to go. And then, of course, genetic testing can then be offered to other family members as well. Having a confirmed genetic diagnosis may not always necessarily predict prognosis and severity. We see a lot of variability. There are a lot of other factors at play as well. So it's not, it gives us with guidance, but it's not necessarily like a crystal ball where we can predict exactly what's going to happen over time. Then the benign or likely benign variants, so that's the other end of the spectrum. This is what we call a negative genetic testing result or unrevealing. No disease-associated variant is identified with a specific genetic testing methodology used in the genes that were tested. So sometimes there's a very simple explanation. The disease-associated gene was not included on the panel. Having a benign variant or a negative genetic testing does not rule out a suspected condition. A child has muscle weakness, even though the genes that were tested for that could explain the muscle weakness were negative or had a benign variant, doesn't change the fact that the child has muscle weakness. So it does mean that the testing that was used did not explain the symptoms. It also does not eliminate the possibility of a genetic or inherited condition. It means additional testing is needed to try and get to an explanation. And then benign or likely benign variants cannot be used to do additional testing for other family members. This means that additional work needs to be done. The really hard or most challenging results that can come back from genetic testing is the variant of uncertain significance. So these can be really tricky to classify and to follow up on. It does not, a variant of uncertain significance in a gene does not confirm a diagnosis. What it means is that there's not enough information to determine if the variant could cause disease. It's important to keep in mind that these variants are very, very common in healthy individuals and in patients as well. So it's hard to tease out whether a variant is associated with symptoms or if it's just a random variant in the information. Then also, the more genes that are being tested, the more variants that will be identified. So usually variants are limited when you do single gene testing, but if you order a whole exome sequencing, most likely a report will come back with many different variants in many different genes, either related to muscle conditions or even completely unrelated to muscle conditions. In all of those situations, additional investigations and additional research is needed. First of all, question to ask is, do the symptoms really fit with what we know is causative, what we know is associated with the reported gene variant? This is when a clinician will go back to the medical literature, or when patients and clinicians will reach out to experts in the field to run the symptoms and run the variant by that person to get their input. And then additionally, what very often also happens, especially in the clinic, is where they would do screening of other family members. If an uncertain significant variant is found in the patient, first step that's typically done is screen mom and dad, see if either one of them carries that variant. If we know that mom carries the variant, then that variant by itself cannot be causative for disease, given that mom's healthy and doesn't have any symptoms. So family screening would then be the first step to do to try and classify these variants. And then also labs and clinicians typically use big population-based databases to see whether these variants are present. We may get really excited about a variant thinking that possibly related to symptoms, but then if we look at these population databases and you know, hundreds of healthy individuals carry the variants, then it may be most likely a benign variant. And then, as I said, these variants may get reclassified over time as additional information becomes available. Sometimes that does require more experimental laboratory-based studies. So in some situation that means extra studies on a muscle biopsy to see if there's that variant of uncertain significance in a specific gene alters the protein of biopsy, or in some situations like with collagen 6 was often done as a skin biopsy to see if there's any changes in the collagen 6 protein in the skin sample. We do not recommend that these variants of uncertain significance are used for clinical decision making. They're variants. We don't know whether they're relevant. They could be relevant, but they could also be a red herring, and we're chasing something, ending up with an incorrect diagnosis label. Well, really, it's a red herring, and there's something completely, there's something else that's actually causative of the symptoms. And also with the variant of uncertain significance, of course, we wouldn't be able to offer carrier screening and family planning within the family. So this is the result that both patients and clinicians struggle the most with, because we just don't know. And hopefully with testing being more available and science catching up, hopefully we're getting better at reclassifying these variants of uncertain significance. But it's now that we're doing more and more exome sequencing, we're also finding more and more variants. We're just starting to sequence genetic information that we didn't use to sequence before. So we're, of course, scientists are learning as they go as well. So how do I get tested, or how does my child get tested? So the genetic testing itself, the procedure is relatively straightforward. It's usually a blood or a saliva sample, sometimes a skin biopsy. It can be done through the neurology or genetics department and clinic. Typical start by clinical-based testing, and then variants come up, or if testing stays unrevealing, there's different options for more research-based testing. Then what should you know before the test is ordered? Hopefully this presentation was helpful. Knowing what methods being used. Are they doing single gene testing, or is it an exome sequencing that's being pursued? And as I said, more is not always better. So testing more genes may not always end up with an easier or faster diagnosis. Then are they doing deletion duplication testing? And then for the exome sequencing, is it just done on the patient, on the child, or are the parents included as well with the analysis? Costs, does insurance cover it? That can vary as well. It can be helpful to have a genetic counselor involved to help with insurance appeals in case of denial. What's the turnaround time, and then what are the possible results? And if there's a variant of uncertain significance, what's being done to try and follow up on the variant to reclassify it over time. Then finally, just quickly, clinical versus research-based testing. So genetic testing can be done most commonly in the clinic. It's ordered through what we call a CLIA certified lab, a commercial lab. There are many different labs around. Many of them have specific panels for congenital myopathies. If that's all negative, sometimes patients and clinicians opt for more research-based testing. There are some important differences. One, of course, is the turnaround time. A clinical commercial labs has a specific process in place and has a specific turnaround time that they have to stick to. Research-based testing, they may operate differently, wait for batches of sample to come in, and send testing off in batches. Their turnaround time may take significantly longer. The regulations are different as well. Clinical labs are CLIA certified labs. They have CLIA regulations to stick to. Research-based testing goes through an IRB research protocol with informed consent that's associated with that. And then the data analysis is very different. The clinical lab uses a set analysis pipeline. They start off and they're focused on known disease-causing genes, and they have a specific threshold that they have to adhere to before they can report back the variants. Research-based testing can be more broad. Usually it's done in a setting with an investigator specifically interested in congenital myopathies or dystrophies. They can take a broader look at the variants, they're more interested in looking at the unknowns, maybe possible interactors, kind of looking outside of the box versus a clinical laboratory. And then the newer technologies are starting to become clinically available. Most oftenly now they're still used on a research basis, so it's the RNA sequencing and then the whole genome sequencing. Still relatively new technologies. With that comes that we haven't really optimized our data analysis yet. They testing generates such a huge amount of data that we really need to develop pipelines still that allow for thorough and efficient analysis. And then are the data for healthy controls, so normal variations seen in healthy individuals for both RNA sequencing and the whole genome sequencing is also still being developed. So that's still something that's coming out, and the more and more data that's available, the easier it is to apply all that knowledge for our specific muscle population. So it's very exciting, but it's still testing that needs to be optimized, and it's not yet ready, completely ready for the clinic. So that's a quick overview of genetics and just genetic testing, genetic strategies for congenital myopathies, both in the clinic and more research-based testing. Hopefully it helps with some of reading through those genetic testing reports. And if there's any specific questions, I'm happy to take them.
Okay, so we're opening it up for questions right now from the audience. If anyone has a question, just go ahead and unmute yourself and ask away.
Yes, Sandra, this is Al Persinger. And you're talking about the different levels of testing from the exome sequencing, the genome sequencing that our son Daniel has been going through. And you said that the exome is like 50% diagnosis. What do you do when there's nothing there and you go up to the genome sequencing and everything, still no specific diagnosis? Is there anything else that one can do, or do you just kind of sit back and keep waiting for science to catch up? Or does it ever help to go back and do another, somebody else to look at the exome sequencing results? Is anything ever found by?
Yeah, no, that's a good question. And unfortunately, it's a situation where a lot of families are in. And yeah, so with the exome sequencing, when all the data is generated, that data remains, that's always available for analysis. So it's not like that's discarded. It's in a big database with all the other patients, and every time either a new finding comes up or a reanalysis is done on a routinely basis, the data is being reanalyzed. So it is an ongoing process, and in some situations it has happened where exome sequencing was unrevealing for years and years and years, and all of a sudden something pops up and we plug it in the database and the answer is right there. It could be as simple as like 1, 2, 3, you enter the data, we enter a new finding and it's there. And that does happen. Then other people looking at the exome data, that's if another team wants to review the data, 2 sets of eyes is better than 1. They may have a different analysis platform or different filters. And it is, that has happened. One team may pick up on a finding that the other team didn't feel was a good lead to follow up on. And then a case-by-case basis, the genome and the RNA-seq is becoming more and more available. With that, again, I don't think we as like clinicians and scientists have really optimized the data analysis. So part of it now is generating the data so we have it, as we're still tweaking all these pipelines. The amount of data and variance that comes back is almost overwhelming for the teams. So we're trying to approach that with using different filters to go through it one by one. So I think that remains to be an ongoing process especially with the genome sequencing. I don't think we can ever say, or at this point we're ready to say, oh, the genome sequencing was unrevealing or normal. It's just that the portions that we looked at didn't provide us an answer, but there's just so much data that we're not good at yet with analyzing. So I think that's an ongoing process. For families, I understand that can be very frustrating. It seems like there's, we've done everything we can do, but on the back end, a lot of the data analysis is ongoing.
Yeah, thank you.
Sandra, there were some write-in questions during your talk, and then some questions that I had emailed to you as well. Would you like to take some time to go over those?
Sure, happy to.
Okay, one question that came in during your talk is, what is your biggest current need to advance your specific research?
Oh, wow. I think biggest need. I think it's a combination of different things, but what has helped us a lot, and I think that's going to help us moving forward, is just like a collaborative approach. The big databases are very helpful. So the NIH has collaborations with the Broad Institute at MIT, and they have developed a really big database of 60,000 data of 60,000 healthy individual, and that's for exome sequencing, and that has made their data analysis really, really helpful because we can easily screen through our variants in the patient and compare it with the healthy individuals. So I think as we're doing more with the RNA sequencing and the genome, it's those collaborative approaches and the generation of big data sets that's going to really help with variant interpretation.
Okay, thank you. And then another question is, what is the best way to find out and get involved in research testing?
I think it's either through discussing it with the local provider. I want to make sure the local clinician is involved, and they can reach out to the various labs. So the NIH is happy to help. Ellen Beck also has a research study for gene identification. So working with the local neurologists to see, they may already know of different studies to be involved in, and then there's clinicaltrials.gov as well.
I think that the Bönnemann lab has been a kind of a go-to place for families who haven't been able to come to a diagnosis for really any of the congenital myopathies or dystrophies.
Yeah, we're happy to help, of course. Yeah, we're absolutely happy to help.
Great. Okay, let's see. There were some other write-in questions. Can you tell based on mutation how severe the disease will be in me or my child?
So it's the mutation. It's not a crystal ball. There's a lot of other factors that come in play. There can be genetic modifiers. There can be just environmental exposures as well, diet, exercise, all of those things. It can tell us in general lines over time what to expect, whether there may be specific additional symptoms that we should think about. Certain mutations and certain genes may have cardiac involvement. So when we have that mutation in hand and we see that there are a number of patients reported with cardiac involvement, we can facilitate appropriate screening and management. In certain situation, we can see how many patients have been reported that had to initiate pulmonary support at a certain age. So it can help guide in general lines specific management and care. But again, it's not it won't be able to predict the future.
Okay, thank you. Another person asked about manifesting carriers. Can you talk a little bit about that?
Oh, yeah, manifesting carriers. So those are commonly, that's with the X-linked inheritance. So we discussed when it's on the X chromosome, the sex chromosomes, females have two X's, males are XY. Typically females, for most of the disorders, wouldn't show any symptoms because they have the backup X that has the normal gene and the normal protein. But in some situation, because of different mechanisms, it's possible that the mother or the female can show some symptoms. That could be either, sometimes it's milder or later onset, or only mild changes, for example, in the CK. So that those are manifesting, female or manifesting carriers of X-linked recessive disease.
Could something similar also happen in other types of inheritance patterns where the individual is a carrier, so they have presumably one healthy copy that maybe just isn't making enough of the protein or not making it correct to the level that the person, experiences no symptoms at all?
Possible. So this is disease specific. So in many of the congenital myopathies, carriers do not have symptoms. For Marfan deficiency, the LAMA2 gene, carriers typically do not have any symptoms, and we know for that gene that 50% is enough for normal function and development. Same for collagen 6 and some of the other like nemaline genes. But there are genes where carriers could have some signs. One of them that we're not really, we don't fully understand this yet, but the Titin myopathies. Titin's a really big and really complicated gene, and there are some reports that in some instances with some mutations, a carrier parent with one Titin mutation may have some cardiac involvement. Research on that still ongoing. But in some, with some recessive diseases, there may be some carrier involvement, but the majority carriers are fine and they don't know that they're a carrier until they have an affected child.
Okay, thank you. Another question that came in from more than one person is about the cause of these mutations. How did these happen in the first place?
It's completely random. There is nothing anyone did or was exposed to or happened during pregnancy, especially these random spontaneous de novo mutations. They just happen. Lots of times these mutations happen. They always happen at conception, and majority of time we don't know about it because they happen in non-relevant DNA, so we never know that these mutations happen. But if a mutation accidentally falls in a specific disease gene, then that can result in symptoms. But it's a random biological occurrence. No exposure, no medication during pregnancy. It's a completely random thing.
Okay, thank you. And there was a fairly long write-in question. I will read it. We did West testing and a single deletion in collagen 6 was found, variants of unknown significance. My husband and mother-in-law have exact same deletion with no symptoms. Currently, we wait on skin biopsy results from NIH. If skin biopsy does not pan out, how can we get involved in further research?
Well, assuming that, I mean, the testing is done at the NIH, so if it doesn't pan out, I'm sure through the NIH there's additional steps that we can do. If that participant can email me, I can follow up on the status of the skin biopsy.
Okay, great. Not sure if she's still on, but Tessa, that was your question. Are there any other questions that others have in the audience? Okay. Well, with that, then I think we will bring this webinar to a close. Thank you so much, Sandra, for taking the time to be with us and share your knowledge, answer our questions. It's been really great. Once today's talk recording has been approved, we would like to post it on the CMDTR website and also other CMD-related websites and social media pages so that those of you who would like to review content or other individuals that just couldn't join us live would have an opportunity to learn from this information. So thank you, everyone, for joining us, and thank you, Sandra.
Thank you. My pleasure.
Bye-bye. Have a great night.
Bye-bye.